The ability of the host to fight the disease-causing organisms is called as the immune system, and that kind of fighting that the body does is called as immunity. Antigens are the foreign substances, so the bacteria, the viruses, the fungi, all those which are trying to attack our body are the antigens. So cellular means these are the barriers or the immunity rendered by certain cells in a body. Hello everyone, a warm welcome to today's session on chapter 8 of 2nd PUC, that is Human Health and Diseases. I'm Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the human health, wherein we discussed what are important for human health, and we also came to know about the different diseases, that is the infectious diseases that affect the human health, and the control measures that can be taken in order to prevent the spread of these infectious diseases. In today's session, let us study about a very interesting topic which is called as immunity. So to begin with, let us start studying with what immunity is exactly. So what is immunity? Immunity is nothing but the response or resistance that is provided by our body to fight against infectious agents. We all know that every day we are exposed to infectious agents, right? We inhale. When we inhale, we take up dust and certain pollutants. Those pollutants and dust will contain a lot of pathogens that are traveling in the air. So every day if we fall ill, then what is the point of living, right? Because every day we'll feel tired, we'll feel very low in energy. So that is the time when our immune cells actually gets activated and fight against the foreign bodies, some of them which are trying to enter into us, therefore trying to keep us healthy and energetic throughout the day. So therefore, every day, since we are exposed to a large number of infectious agents, wherein only a few of these are the ones that causes diseases. So it's not that our body is immune to cholera, dengue and all that. No, it's not like that. So there are some diseases which our body is not immune to, wherein we will contract those diseases, and therefore we'll have to go to the hospitals to take medicines for that. But there are some diseases, say for example the common cold. Not every day we suffer from the common cold. Every day we are exposed to the common cold virus, right? But do every day we suffer from common cold? No. It's only during certain times when our body is low in immunity, that is a time when we suffer from a common cold. So therefore, against some of the infectious agents, our body is capable of providing immunity against them. So this is due to the fact that the body is able to defend itself from most of these foreign agents, and this overall ability of the host to fight against the disease-causing organisms is called as the immune system, and that kind of fighting that the body does is called as immunity. So therefore, immunity is provided by the immune system of a body, which produces certain type of immune cells that actually render protection to our body against some of the infectious agents. So immunity therefore is of two types. One is innate immunity and the other one is acquired immunity. So what are these? Let's look into. So first, learning about innate immunity. So innate means it is there within us. So this type of immunity is there in our body right from the time we are born. So that is why it is called as innate immunity. So innate immunity, it is a non-specific type of defense that is present at the time of birth, and this innate immunity provides different types of barriers, or it results in the formation of different types of barriers that prevent the entry of foreign agents into our body. And this immunity again innate immunity consists of four types of barriers. So barriers means something which prevents something, right? So here innate immunity is a type of immunity where the immune cells act as a barrier. Those immune cells which are not specific to a particular disease, but generally they are specific to a number of infectious agents and which are present in a body right from the birth itself, and they act as a barrier against certain infectious agents. What are the 4 types of barriers the innate immunity gives us? Let's look into. So the first one is the physical barrier, the physiological barriers, the cellular barriers, and the cytokine barriers. So let's study about each of these one by one. Innate immunity, the first type of barrier is the physical barriers. So physical barriers means something which you can touch, something which you can feel. So the best physical barrier that is present in a human body is the skin. So the skin in our body is one of the main physical barrier that covers our entire body, that prevents the entry of the microbes into the body. And apart from that, there are also some other physical barriers that are present inside a body as well. Say for example, we have the respiratory system. So our nose, for example, it is coated with mucus. Why it is coated with mucus is because when we breathe in air, that air would contain a lot of infectious agents that should not enter into a respiratory tract and cause diseases or respiratory disorders. So to prevent that, inside our nose, there is a layer of mucal cells that are present. Just like that, the epithelium lining of the respiratory, the gastrointestinal tract, and the urogenital tract. So respiratory tract means our trachea, our nose, nostrils, everything are coated with mucus lining. Gastrointestinal tract means our esophagus, the intestine, everything is coated with mucus lining. And urogenital tract, that is the urinary tract, that is also coated with mucus lining, so that because through these openings the infectious agent should not enter into the human body. So that is why. And these mucus coatings actually, since they are slimy and sticky, they help in trapping the microorganisms that are trying to enter into a system. So that is why the 2 important types of physical barriers under innate immunity are the skin and the mucus coating of the respiratory tract, the gastrointestinal tract, and the urinary tract. So next, moving on to the physiological barriers under innate immunity. So physiological is something related to biochemical. So what are the different biochemicals that are produced in a body? Say for example, in our stomach. So when we eat food, what happens? In that food, there might be some microbes that are there. So that microbe may cause stomach flu, may cause food poisoning, and a lot of things. But our body has the capability of providing innate immunity in the form of producing some physiological barriers, and one such is present in the stomach, which is made up of an acid, that is HCl. So therefore, the acid in the stomach, the saliva in the mouth, the tears from the eyes, all these prevent microbial growth. So sometimes you might have heard the old people or during the old times, or your mother, sometimes she might say, please cut an onion. Occasionally, whenever you have time, cut an onion, so that when you cut an onion, your eyes will start to water, and through that eye water, all the impurities and microbes and all will come out. So it will clear the eyes. So that is why eyes get cleared in the form of water. So when something enters into your eyes also, your eyes will start to water. Why is it? Because so that whatever irritant has entered into your eyes, it has to come out through the water. So that is why the tears from the eyes are also a physiological barrier here. Next, talking about the cytokine barrier. So cytokine barrier, cytokines are nothing but it is a kind of protein that is produced in the body. These proteins actually, say for example, there are some virus infected cells. So whenever a body is infected by virus, we all know that there is no cure for virus, right? no cure in the sense, because the virus will have a certain life cycle, and until that life cycle of that virus in our body gets completed, the infection will be there whether you take a tablet or not. Only thing is you get medications to control your symptoms so that you can do your daily activities very well. Apart from that, viruses usually, there are no medicines to kill a virus. Like unlike we have antibiotic, antifungal agents and all that, as such for virus it is not available. So therefore, those cells which are infected by virus, they secrete some proteins which are called as interferons, and these interferons protect the non-infected cells so that the virus do not attack the non-infected cells. Say for example, viruses, they have entered into the body and they are causing a disease. So what viruses do? We all know that viruses are capable of multiplying only when it is inside a host organism or only when it is inside a body, right? So now when it has entered into the body, those viruses will start to multiply. When they start to multiply, they will not just remain in one cell. They will start to enter into other cell, spreading the disease eventually to entire body, right? So that is the time to protect before the virus starts to divide and infect other cells that are there in the body. Before that itself, what happens, those cells, virus infected cells, will start to produce certain proteins which are nothing but cytokines, and they are called as, those proteins are called as interferons. What these interferons will do is, they will protect those cells which are not yet infected by the virus, therefore preventing the spread of infection to the healthy cells in the body. So they are the cytokines, and cytokines get activated usually when the body gets infected by a virus. Next, talking about the cellular barriers. So cellular means these are the barriers or the immunity rendered by certain cells in a body. So here certain types of cells such as the leukocytes. So leukocytes are also called as the white blood corpuscles or white blood cells, and they are the ones that actually provide immunity to our body, right? So there are certain types of leukocytes in a body, like the polymorphonuclear leukocytes, which are called as PMNL neutrophils, and there are certain monocytes which are present, and there are certain types of lymphocytes that are present which are the natural killer. So all these which are there in the blood, so all these are the different types of WBC itself. So among the different types of WBCs or leukocytes that are present, three important types of WBCs, that is polymorphonuclear leukocytes or WBC, monocytes and lymphocytes. They actually protect our body against the macrophages. So macrophages are nothing but those organisms which have entered into the body from outside and are trying to feed on to our cells. So they are called as macrophages. So what they do is, once they start to try to infect the blood and feed on the blood cells, these three types of leukocytes, they get activated and they will start hunting on to these particular macrophages, and they actually render a mechanism which is called as phagocytose. So phagocytose means, like phagocytose is seen in amoeba also, how amoeba engulfs its food, right, by moving near its food. Just like that, these leukocytes, different type of leukocytes also, they just detect that particular foreign body or the macrophage and they try to engulf it, and they will just take them inside and completely destroy the macrophages, therefore destroying the microbes as well. So microbes are nothing but the macrophages as well here. These are the cellular barriers, and one of the best example is the three types of leukocytes that I have mentioned here. So next, moving on to the second type of immunity, that is the acquired immunity. So acquired immunity is something that we acquire later on. So as I told you, understand the difference here. Innate immunity is not specific to one kind of a pathogen or one kind of a infectious agent, because it is generally produced in a body right from the birth, whether we have an infection or not, whether an infectious agent is trying to enter into a body or not. Innate immunities or immune cells are already present in a body during childbirth, but acquired immunity is not present during childbirth. Say for example, acquired immunity is produced whenever we are trying to get a disease. Say for example, common cold. When you get a common cold, what happens? Or when that influenza virus enters into your body, at that time the new immune cells will start to be produced. It will not be there before itself. When you're having an infection, at that time the immune cells will start to get produced. That is called as acquired immunity. So acquired immunity is always pathogen specific, and they are specific. Say for example, acquired immunity, we can say for the coronavirus, our body now has acquired immunity, right? From where did we get that acquired immunity? We got it from the vaccination. So that immune cells are specific to coronavirus itself. When if for other common cold viruses, it is not specific. If common cold viruses come, then different type of that is different type of immune cells need to get produced there. So that is about acquired immunity, and it is characterized by memory. Say for example, we have a disease. So for that particular disease, say typhoid. Okay, for that typhoid which is caused by the bacteria Salmonella typhi, for that some immune cells are produced in a body. Now that is acquired immunity. Was it present during the birth itself? No, it was not present. When did it get formed in the human body? It got formed in the human body when that person got affected with or infected with Salmonella typhi bacteria. So when that person got infected with Salmonella typhi bacteria. So when Salmonella typhi bacteria infects the person, for that particular Salmonella typhi bacteria which is causing typhoid, for that only the immune cells will be produced. Now that is acquired immunity. So and next time, so now the immune cells have been produced, right? So next time, say for example, after 2 or 3 years, again that person gets typhoid. Now the memory will be in such a way, the person's body cells will memorize the bacteria, and automatically it will start producing the immune cells, because it knows that here this person was infected by Salmonella typhi. Already the immune cells have been produced to that particular Salmonella typhi, and therefore the immune cells will be memorized and will be stored like that. So therefore it is characterized by memory of the cells, and this means when a body encounters or when into a body a pathogen enters for the first time, it produces a response which is called as primary response, and it is of low intensity. So as I told you, first time when Salmonella typhi infects a person and immune cells are produced, it is called as primary response. And subsequently after 2 or 3 years, again if Salmonella typhi infects the person, then it is called as secondary response. Why did that secondary response get produced? It is because of the memory that is stored in the cell. So that is why. So subsequent encounter with the same pathogen, it will increase a high response, immune response, which is called as secondary response or anamnestic response. So anamnestic response or secondary responses, second time production of immune cells to the same pathogen when it affects the person. So this is ascribed to the fact that our body appears to have a memory of the first encounter. So that is how, like how we have coronavirus, right? When was the vaccination given to us? Right, but still if we get coronavirus, of course booster dose is there. After several months we have to take a booster dose, but apart from that those immune cells will already be present in our body. So next time again when you get coronavirus, the virus will infect our body, but the symptoms it causes will be very, very mild. Why? Because it is secondary response that the immune cells are providing to the body. So that is because of the body's memory or the cell's memory that is there. So this is about acquired immunity. So therefore the primary and secondary immune responses are carried out by two special types of lymphocytes which are present in our blood, that is the B lymphocytes and the T lymphocytes. B lymphocytes and T lymphocytes are very, very important, and they are present in the blood. They are the components of the white blood cells as well, and they are the ones that give us the acquired immunity, because they are the ones that get produced. There are different diseases specific B lymphocytes and T lymphocytes will be there. So B lymphocytes, as the name suggests, it is produced by the bone marrow. So that is why it is called as B lymphocytes. T lymphocytes are produced by the thymus gland. So that is why it is called T lymphocytes. So B for bone marrow, T for thymus there. So let's study about these lymphocytes under acquired immunity. So why are they acquired immunity? Because these lymphocytes are produced only when a pathogen attacks. Because to that specific pathogen only these lymphocytes will be produced. And it is not like the B lymphocytes produced for typhoid will also produce the same lymphocytes for jaundice as well. No, it is not like that. For typhoid, different lymphocytes are produced, different cells are produced, immune cells. For jaundice, different immune cells, because they have specific shape and structure wherein they can act as phagocytic cells and engulf the foreign bodies that are trying to enter into. So that is why they are specific to a specific disease always. So the B lymphocytes usually they produce an army of proteins, that is they produce different sets of proteins in response to pathogen in our blood to fight with them. So that is why whenever we have an infection, and if you go and get your blood test done, you will notice that the WBC count, especially the lymphocytes count, would have increased, stating that the body is infected with the disease and our immune cells are working against fighting the diseases. So that is the time when these lymphocytes actually get produced. So these proteins which are produced are called as antibodies. Anti means against. Against what? Against a foreign pathogen. Against a foreign substance that is entering into our body and causing diseases. So therefore, these proteins are called as antibodies. So whenever a foreign substance or whenever a pathogen enters into our body and tries to cause diseases, for that particular pathogen, specific antibodies will get produced. So what are these antibodies? Antibodies are nothing but the proteins that are produced by the B lymphocytes, which are a part of the acquired immunity. And the T cells which are there, or the T lymphocytes, they do not secrete any antibodies. But what is their function? They also render immunity to our body, but here they are saying that T lymphocytes or T cells do not produce any antibodies. In spite of that, what is their function? Their function is to help the B cells to produce antibodies. So therefore, if the B cells need to produce antibodies, if the B lymphocytes have to produce antibodies, then it should take the help of the T lymphocytes. That is why B lymphocytes and T lymphocytes both act as acquired immune cells, or the immune cells which are a part of the WBC. Next, let's study in detail about the antibody. What is this antibody? So each antibody molecule, especially the structure when we consider, it has 4 peptide chains. As you can see here, it has 4 peptide chains. So 1, 2, the third one and the fourth one, right? So 4 peptide chains are present. So in that 4 peptide chains, 2 chains are smaller. You can see here 2 chains are smaller, right? So these are small chains, one here and here. And since they are small, they are light in nature. Therefore, they are also called as the light chain. So these 2 chains, they are also called as light chain because they are lighter in appearance. So that is why. And apart from these, 2 chains are longer chains. So these are the longer chains. This one also. And therefore, they form the heavy chain. So what is the structure of the antibody usually? It is made up of 4 peptide chains. In that, 2 chains are shorter and they are called as the light chain. 2 chains are longer and they are called as the heavy chain. And hence the antibody is represented as H2L2, meaning 2, H means heavy, L means light. How many chains are there totally? 4. Out of that, 2 is heavy. Therefore, H2, 2 is light, therefore L2, and hence the structure of the antibody is represented as H2L2. So there are different types of antibodies that are produced in a body, and some of them are given here. Let's look into them. So first one is IgA, that is immunoglobulin A. Then we have immunoglobulin M, immunoglobulin E, and immunoglobulin G. So these are the different types of antibodies. There are various others also, but in your textbook they have mentioned these 4 antibodies, that is immunoglobulin A, M, E, and G are the different types of antibodies that are there. So different types of antibodies that are there, and these antibodies have a specific defense. So some forms the first line of defense, some form a defense against a particular pathogen, or some form a defense against only a particular parasite. So like that, they are particular in their function. And these antibodies are found in the blood. So they are found in the blood, and the response of these antibodies are also called as humoral immune response. So they are also called as humoral immune response because they are the immune cells produced in the plasma of the blood. So plasma, we all know that it is a colorless fluid, right? So therefore it is also called as the humoral immune response. So moving further, let's study about the types of antibodies. So what are the different types? Types of acquired immunity. So types of antibody we have studied: IgA, IgE, IgM, and so on. Types of acquired immune responses. So there are different types. One is antibody mediated. Antibody mediated means for a specific pathogen, a specific antibody will be produced when the disease strikes to that particular person, and therefore the person can be cured of that particular disease, or the symptoms can be curbed to some extent. So that is antibody mediated. Next is cell mediated immune response, or it is also called as cell mediated immunity. So cell mediated immunity, say for example, as I told you, how for a particular virus, how the cells act providing immunity so that further damage cannot occur. Next is the T lymphocytes mediated cell mediated immunity. So the T lymphocytes mediated cell mediated immunity are provided by the T lymphocytes. So what are T lymphocytes? They are the lymphocyte cells or the immune cells that are produced by the thymus gland, and they do not have a specific function, but their function is to help the B lymphocytes produce the antibodies. So therefore, those cells which take the help of T lymphocytes to produce antibodies are called as T lymphocytes mediated cell mediated immunity. And very often when some human organs such as the heart, eye, liver, kidney, whenever it fails to function, that time transplantation is the only option that is left. So when organ failure occurs, none of the immune cells can bring the organ back to life. So in such conditions, only organ transplant can be done. So when an organ transplant is done also, that time what happens now? Say for example, there is 2 persons, one who is a patient and the other one is the donor. So that patient's heart has failed and nothing can be done now other than transplanting the heart. So a donor will come, or there will be a donor. For some reason that person might have died, but his heart is in good condition. So what doctors will do is, everything matches, then the doctor will remove the donor's heart and he will put it into the patient. Now, from where did the heart come? It came from some other person, right? Does the heart belong to the patient? No, it came from some other person. So what a body will start to think is, it is a foreign agent. I need to attack it. So therefore it will start producing immune cells to attack that particular heart. So at that time what happens is, in such patients immunosuppressants will be given so that the immune cells will be reduced in the body. Therefore those immune cells cannot act on the particular heart that is transplanted. So therefore transplantation is one of the best method in patients who have an organ failure. Let us talk about two types of immunity. So one is active immunity and passive immunity. So active immunity, when a host is exposed to antigens. So what are antigens? Antigens are the foreign substances. So the bacteria, the viruses, the fungi, all those which are trying to attack our body are the antigens. So when a host is exposed to antigens, which may in the form of living or dead microorganisms, or it may be in the form of proteins, that is viral proteins, or it may be in the form of proteins like the viral proteins and all that. At that time, antibodies are produced in the host. So this type of immunity is called as active immunity. So active immunity becomes active whenever a external organism or an antigen or a protein, etc., tries to enter into a body. Antigen in the form of a living or a dead microorganism, or in the form of protein, when it tries to enter into a body, the immune cells get produced or antibodies will get produced. So such type of immunity is called as active immunity. And active immunity is very slow. Why it is slow? Because it is not, this is a type of acquired immunity. Why? Because unlike the innate immunity which is produced during the childbirth itself, these during active immunity, the acquired immune cells that are there, or which it is, because it is a type of acquired immunity, when the pathogen attacks, the body cells should recognize the pathogen, or the immune cells should recognize the pathogen, and it should produce antibodies respective to that particular pathogen itself. So to do so, it will take a lot of time. So therefore, active immunity is slow and it takes time to give its full effective response. So it takes a lot of time and also it is slow. Injecting the microbes deliberately during immunization or infectious organisms gaining access into the body during natural infection induces active immunity. So the best example that I can take here of injecting the microbes deliberately. The best recent example that we can take of is the coronavirus, right? Wherein different types of vaccines have been produced. We have Covishield, we have Covaxin, Sputnik, various types of vaccines are available, right? Pfizer, so different types are there. So what do these contain? They contain either, they contain the dead microbes or the inactive microbes. Some of the vaccines contain inactive coronavirus itself. Some of them contain the protein of that particular virus. So what happens here is, before our infection, say for example, I am a healthy person, I have not got infected with corona. If I was infected with corona, then already my body would have started producing immune cells for that. Now I have not been infected from corona, I don't want to get infected by corona, so I'll have to take a vaccination. Now how will a body produce immune cells? Because I have not got any corona infection. And my body should know what type of immune cells it has to produce, what type of antibody it has to produce, right? So that is the time vaccines will be designed in such a way, either it will contain a inactive or a dead coronavirus itself, or it will contain a protein of that coronavirus. Now that will be given into my body. So as soon as it enters into my body, it will not cause any infection to me. Why? Because it is dead, it is inactive, right? But that content is there in my body now, right? My body will start to produce immune cells, or it will start to produce antibodies which will fight against that particular coronavirus. Next time when coronavirus actually infects my body, already my body's memory would have been activated and will start producing the same type of antibodies against coronavirus. So that is how the injecting the microbes deliberately means. It is not to let the person get disease, but it is to make the person immune to that particular disease whenever he gets that disease. So that is why. So all these are different examples for active immunity. Next, talking about passive immunity. So passive immunity is also a kind of acquired immunity when ready-made antibodies are directly given to protect the body against foreign agents. So it is called as passive immunity. So passive immunity means our body will not produce any kind of immune cells for that particular pathogen or for that particular antigen. So what doctors will do is they will design a vaccine or something and deliberately inject it into a body. So they are ready-made antibodies we have various antibiotics, we have various antifungal creams, all that available, right? So like that, they're deliberately given to a body. They are ready-made antibodies which are given to a body so that it will help us fight against the foreign agents, and therefore it is called as passive immunity. So one of the best example here for passive immunity is, passive immunity can be artificial or it can be natural as well. So one of the best example is, we have colostrum. So what is colostrum? You all have studied about this in human reproduction chapter, right? Wherein the mother, once she gives birth to the child, the first milk that comes out of her mammary gland, it is yellow in color and it contains lots of antibodies, and that yellow color milk is called as colostrum. And that colostrum that is secreted by the mother during initial days of lactation, it will contain abundant antibodies, that is immunoglobulin A. And feeding the child with this particular colostrum will indirectly, the antibodies will go into the child. Now the child has acquired passive immunity, right? Because the immunity is got from somewhere else. It got it from the mothers through mother's colostrum, which is the first milk that comes out of the mammary glands. So therefore, it can be maternal as well. And the fetus also receives some of the antibodies from their mother, that is through the placenta during pregnancy. So during pregnancy also, through the placenta, some of the antibodies will enter into the fetus so that any infection we get, say for example, we get common cold, that common cold or the influenza virus should not affect the fetus as well, right? So therefore, some of the antibodies through the placenta, it will go to the fetus also. So this is a best example for passive immunity. So this is a best example for passive immunity, and this is a natural passive immunity. Other one is artificial one also, as I told you, which are artificially made in the laboratories, and those antibodies are given to the patient so that their body can start fighting against the infectious diseases. So this was about the topic, that is today's topic, immunity, wherein we studied about two types of immunity, acquired immunity and innate immunity. Under acquired immunity, we studied about the active immunity and the passive immunity, and also we studied the structure of the antibody. So I hope you understood this session well. In the coming session, let us deal with vaccination and immunization to protect ourselves from different diseases, and also we shall study about allergies and autoimmunity, that is the immunity that is produced in the body. So with this, we shall move on to the next topic in the next session. So we shall meet again in the next session. Thank you.